Hello everyone, welcome to my presentation. Before we begin, I'd like to thank everyone involved in this conference for giving their time and effort to make this happen. I would also like to thank my fellow co-authors, in particular my research supervisor, Trevor Hamilton. To start, I'm going to share some relevant background information related to our study. Recirculating aquaculture systems, or RAS for short, are essentially a technology used in aquaculture to farm fish. One of the benefits of these systems is that the environmental conditions within the system can be precisely controlled and regulated. Our study investigated the effect of photoperiod and salinity on the behavior of coho salmon that were reared in RAS. We examined the behavior of these fish because it can reflect overall fish well-being and can potentially identify suboptimal rearing conditions. We assessed behavior using 3 different behavioral tests: the open field test to quantify locomotion, the novel object approach test to quantify boldness behavior, and the light-dark test to quantify anxiety-like behavior. The coho salmon used in this study were reared by our collaborators at UBC for 120 days in RAS on either a 12-hour photoperiod or a continuous 24-hour photoperiod, and at salinities of 2.5 or 10 ppt. An additional group was also reared under 24-hour light and 30 ppt. If we take a look at the photos on the bottom right area of the poster, you can see the RAS and coho salmon that were used in this study. Behavioral testing began with the open field test, where fish were placed into a circular arena and distance moved and meandering were measured for 5 minutes. Afterward, we introduced a multicolored novel object in the center of the arena for the novel object approach test, where the time spent in the thigmotaxis zone, which is the outer zone closest to the walls, the transition zone, and the inner zone of the arena was measured for 5 minutes. Fish were then transferred to the light-dark test arena, where time spent in the dark zone was measured for 15 minutes. Now moving on to the results section. In figure A, we can see 2 different visualizations of fish movement. The photos with only red lines are called track plots, which trace the fish's movement in the arena. The other 2 pictures are called heat maps, which reflect the localization of fish in the arena throughout the trial. Areas represented by longer wavelengths like red indicate more time spent by the fish in that area than areas represented by shorter wavelengths like blue. In figure 1, B, and C, for the open field test, we observed no significant differences for distance moved and meandering, which suggests that photoperiod and salinities investigated did not have a significant impact on locomotion. Results for the novel object approach test are seen in figures D, E, and F, where we observed no significant differences for time spent in the inner zone. However, the 2.5 ppt 12-hour group spent significantly less time in the transition zone than the 10 and 30 ppt 24-hour groups. The 2.5 ppt 12-hour group also spent significantly more time in the thigmotaxis zone relative to the 10 and 30 ppt 24-hour groups, suggesting that it was less bold because it spent less time inspecting the novel object. Moving on to the light-dark test in figure G, we observed that the 2.5 ppt 12-hour group had a preference for the light zone. This would normally indicate decreased anxiety-like behavior. However, there are no significant differences in time spent in the dark zone between any of the groups, suggesting that anxiety-like behavior may have only been minimally affected. In figure 1H, it shows the track plots and heat maps for 2 of the groups in the light-dark test, so you can see what the arena looked like. Lastly, to make sure that fish size was not a confounding variable in these behavioral tests, we also measured the fish's weights and lengths and did not find any significant differences between groups. Prior to collecting data, we hypothesized that the fish reared under 24-hour light in RAS would demonstrate significantly different behavior on these behavioral measures than those reared under 12-hour light. Contrary to our hypothesis, and what we believe is the most important and interesting finding from this study, is that overall, coho salmon reared under a 24-hour photoperiod did not demonstrate any negative locomotion, boldness, or anxiety-like behavioral effects compared to those reared on a 12-hour photoperiod. Our study provides support for the use of a 24-hour photoperiod for rearing coho salmon in RAS, as we have shown that it has minimal impact on behavior. Thank you for your time. That concludes my presentation.